Good morning, students. Today we will start new chapter, that is biological classification. Aristotle's classification. Aristotle was the earliest to attempt a more scientific basis for classification of organism. He classified plant into trees, shrubs, and herbs, and animals into two groups: those which had red blood and those that did not. In the first diagram, you can flow chart, you can see animals that with red blooded animals, that is mammals, lizard, bird, and fish. Where no red blood, that is hard bodies insect, soft bodies insect, snails, shellfish, jellyfish, such kind of examples. Next is two kingdom classification that were proposed by Carl Linnaeus in 1758. The system classifies organism into two kingdom, that is plant and animalia. Now after this, what was the drawback of two kingdom classification? The prokaryote, that is bacteria or cyanobacteria, eukaryotes, fungi, mosses, ferns, gymnosperm, angiosperm were included under plant. It is based on the presence of cell wall, but prokaryotes and eukaryotes are widely differ in other features. It include unicellular and multicellular organism in the same group. Spirogera and Cladomonas were placed under algae. It did not differentiate between the heterotrophic fungi and autotrophic green plant. Fungi have chitinous cell wall while the green plant have cellulosic cell wall. These are the basic drawback of two kingdom classification. First is what? Prokaryote and eukaryote were in the same group. Unicellular, multicellular were with the same group, that is. And second, it did not differentiate between heterotrophic and autotrophic plant. They were in the same group. So these two drawback led to the foundation of five kingdom classification by R.H. Whitaker, 1969. It include Monera, Protista, fungi, plantae, and animal. This classification is based on cellular structure, thallus, organization, mode of nutrition, reproduction, and phylogenetic relationship. First kingdom is Monera, kingdom Protista, kingdom fungi, kingdom plantae, kingdom animalia. This is the five kingdom classification: Monera, Protista, fungi, plant, animalia, and their characteristic: cell type, cell wall, nuclear membrane, body organization, mode of nutrition. Based on all this, the basic five characteristic of each kingdom, five kingdom is given here. Cell type: Monera, that is prokaryotic; Protista, eukaryotic; fungi, eukaryotic, means as you go to the higher level, only Monera prokaryotic, rest of all are eukaryotic. With the help of this flow chart, it is also easy to learn. So better you can make it in your copy. Now, Kingdom Monera, that is bacteria. As you know very well, they are unicellular, prokaryotic, sometimes form a group or filaments. Bacteria are most abundant microorganism. Hundreds of bacteria are present in a handful of soil. They also live in extreme habitats such as hot spring, deserts, snow, and deep ocean. Many are parasites. All the bacteria are not parasite. Some are also very useful. For example, Rhizobium bacteria, which is very helpful to plant, it converts free nitrogen into nitrate and nitrite. And many bacteria which are also present in our body, that is very helpful. For example, that bacteria present in milk, that is lactobacillus. Based on the shape, bacteria are divided into four categories. Means that if the shape is spherical, that is coccus. If the shape is rod shaped, that is bacillus. You can easily see this figure and understand. Comma shaped, that is vibrium, vibrospores. Easily. Last is spiral, spiralum. You can see this diagram. Bacterial structure is very simple, but they are complex in behavior and show metabolic diversity. Some bacteria autotrophic, synthesize food from inorganic substance. Majority are heterotrophic. They do not synthesize the food but depend on other organism or dead organic matter for food. This is the structure, that is capsule, cell wall, plasma membrane, cytoplasm, rhizobium. This is the flagella, this part that help in the movement of that bacteria. This is the classification of bacteria. Archaeobacteria and eubacteria. Archaeobacteria is further divided into halophiles, thermoacidophiles, methanogens. Now, eubacteria are further divided into autotrophs and heterotroph. This autotroph is further divided into photosynthetic autotrophic, chemosynthetic autotrophic, and this heterotrophic are further divided into parasitic and saprophytic. Means that eubacteria is divided into autotrophs, means that they prepare their food with the help of their own. First method is through that photosynthesis, and second is through chemicals. And heterotrophic means they are dependent upon other for their survival, means that bacteria are so parasitic and saprophytic. Next is archaebacteria. They live in hard set habitats such as extreme salty area. Halophiles means halophiles are the area which contain high amount of salt. Hot spring, thermoacidophile, and marshy area, methanogens. Archaeobacteria have a different cell wall structure for their survival in extreme conditions. Methogens are present in the guts of ruminant animal, that is cow, buffalo, etc. They produce methane biogas from the dung of these animals. Eubacteria, that is true bacteria. They have a rigid cell wall and a flagellum if motile, means flagellum is for their movement. They include autotrophs, that is photosynthetic and chemosynthetic, and heterotrophic. Photosynthetic autotroph, that is cyanobacteria. They have chlorophyll A. Chlorophyll is also divided into four types: chlorophyll A, B, C, etc. Similar to that of green plants. Cyanobacteria, that is blue-green algae, it is also with other name. Unicellular, conical, filamentous, marine or terrestrial algae. The colony are generally surrounded by gelatinous sheet of viscous substance. They often form bloom in polluted water bodies. Some of them fix atmospheric nitrogen in the specialized cell called heterocysts. Example, nostoc and Anabena. This is the structure. Heteroset. Mucilaginous sheet is there. This is the example of Nostoc bacteria. This is Anabena. That structure you can see. Other structure of few bacteria. Chemosynthetic autotroph. They oxide inorganic substances such as nitrate, nitrite, ammonia and use the released energy for ATP production. They play a great role in recycling nutrients like nitrogen, phosphorus, iron, and sulfur. Thanks for today. Tomorrow we will proceed further.